Hello, welcome to EPG Pathshala. The paper is Methodology of Research in Sociology. I am Dr. Sudarshana Sen, Associate Professor, School of Women's Studies, Jadavpur University. The number of this module is 14 and name of the module is Research Methods for Testing Procedures. In this module, we will be talking about experimental research design. Experimental research design is not very popularly used in sociology, though this has been in use in the last 50 years. Experimental research design aims at testing a hypothesis where we try to find out cause of a phenomenon. Experimental research is based on testing a hypothesis stating a relationship between a dependent and an independent variable. Experiments are the best method in social science for establishing causality. The four elements of an experimental research design are manipulation, control, random assignment and random selection. Manipulation means something is purposefully changed by the researcher in the environment. Control is used to prevent influence of extraneous variables. Random assignment means that if there are groups or treatments in the experiment, participants are assigned to these groups or treatments or randomly, or that no matter whom the participant is, he or she has an equal chance of getting into all of the groups or treatments in an experiment. Random selection is how the researcher draws the sample of people for the study from a population. Suppose X is present in situation A and Y is also present and in situation B. It so happens and corroboratively, whenever there is an absence of X, Y is also absent, we can infer that there is a relationship between X and Y and that the relationship is of a causal nature. It means that X is the cause of Y and that whenever X occurs, Y follows and not vice versa. First, such logic in research helps to rule out various factors that may occur together. Second, it helps us to recognize that two or more factors may occur together. Third, it allows us to observe that in concrete situations, X always occur prior to Y. Now, it can simply follow from this that whenever X occurs, Y follows, which leads us to formulate a design where we will try to prove that X is the cause of Y. The second question, how is an experimental design executed? The experimental logic in its pure form can be executed in a laboratory setting. In order to do an experimental study, the researcher starts with a hypothesis. In order to test this hypothesis, the experimenter must establish two groups, the control group and the experimental group. Each group is matched in every possible way. They will be put into identical environments which are controlled in every respect by the experimenter. In order to test this hypothesis, the experimenter must establish two groups, the control group and the experimental groups. Each group is matched in every possible way. They will be put into identical environments which are controlled in every respect by the experimenter. The experimenter will then deliver a certain amount of stimulus to the control group, but will systematically vary the amount of stimulus delivered to the experimental group. If there is variation in the experimental group and this variation can be linked with the variations in the stimulus, then the experimenter can assume that there is a causal link between the stimulus and the result, since there is no other difference between the two groups. The variable that is controlled by the experimenter is called the independent variable X, and anything that varies as a result of this is called dependent variable Y. Causality tends to be searching for a single cause of a single event. But we know from common sense and scientific thinking also confirms that there are necessary and sufficient conditions that also influence an event. Scientific explanations differ from common sense in way of explaining that there is a single cause of an event. Scientific explanations take into account not only conditions in which one identifies stimulus can be present, but also contributory conditions, contingent conditions, alternative conditions, all of which are expected to be operating upon the effect. A necessary condition is one which must occur to be the cause of an event. A sufficient condition is one which is always followed by the effect. A condition may be both necessary and sufficient for the occurrence of a phenomenon. A contributory condition is one that increases the probability that a given phenomenon will occur but does not make it certain. This is because it is one of the many factors together which determines the effect. A contingent condition is one under which a given variable is a contributory cause of a given phenomenon. An alternative condition is that which may make occurrence of a phenomenon more likely. These help us to understand that no phenomenon can occur due to a single cause, or putting it in a different way, it can be said that there are multiplicity of causes behind the occurrences of any phenomenon. This helps us to understand that no phenomenon can occur due to single cause, and to do this in real life, the researcher has to select a population appropriate for the hypothesis to be tested and divides a sample to be studied into two equally representative groups. There should not be significant difference between the two groups except the experimental variable. Both the groups will be given a similar test. Say for example, the researcher wants to know the attitude of people towards nuclear electric power stations in neighborhood. From the results of both the groups, the researcher will match the response of the test and arrive at a conclusion on which is the factor that may signify the difference. The factor X identified will be the cause of the difference Y. The ways in which the two tests are evaluated are precision making, matching by frequency, distribution, and by randomization. In precision making, every element in the control series will be represented with the same qualities in the experimental series. To find out individuals in the control series with the same qualities in the experimental series is difficult in practice. In matching by frequency distribution, the researcher seeks to have equal representation of relevant factors. In randomization, the researcher starts with the assumption that half of those having a particular characteristic will fall in each group, even though the researcher may discover only later that a certain variable is significant. There are problems with the classical experimental design that I have been presenting so far. First, being the problem with recognizing and controlling the variables. Control here in practice means that the values of the variables are or may be deliberately minimized to achieve desired results. The second problem is that the causal relationship may not be clearly stated. The third problem is the element of time. Any social variable requires time to affect social behavior. If tests are carried out immediately after the stimulus, the change is less likely to be observed. The fourth problem is that the classical design is stated in a very simplistic manner. It is stated in a quantitative form. Through the statistical technique with the use of probability theory, it may be proved that Y is the cause of X. With the use of better techniques of measurement like concomitant variation, for example, this may become more easy. Now, there are some deviations from experimental designs. So, the classical design though paved the road for experimentation in social science, there are various other types that have been formulated at present. Before-after experimental designs. In such designs, a single group is selected and the dependent variable Y is measured before the introduction of the experimental variable. The effect of the treatment is level of phenomenon after the treatment Y minus the level of phenomenon before the introduction of the treatment X. After only experimental designs. In this design, two groups are selected and the treatment or the stimulus is introduced into the experimental group only. The effect of the dependent variable is measured at the two groups, but at the same time. The impact of the treatment is assessed by subtracting the value of the dependent variable in the control group from its value in the experimental group. Here the assumption is that the two groups, control and experimental, are identical with respect to their behavior towards the experimental variable. Before-after experimental design or successional experimental design. In this study design, two groups are selected and the dependent variable is measured in both the groups, control and experimental, for an identical time period before the treatment has been introduced. The treatment or the stimulus is introduced only in the experimental group and the dependent variable is measured in both the groups for an identical time periods. The effect of the treatment is measured by subtracting the change in the dependent variable in the experimental group or situation. Completely randomized experimental design or CRD. The characteristic of this design is that elements are randomly subjected to treatment or vice versa. It provides maximum degrees of freedom to the error. Such designs are used when the experimental areas is homogeneous, or technically speaking when all the variations due to uncontrolled extraneous factors are included under chance variation. It is characterized by two principles, replication and randomization. In a randomized design, a sample is selected randomly from the population and randomly identified as control and experimental groups. The two groups are given two different treatments and each group is tested before and after the treatment is introduced. The results are compared and the findings help to either accept or reject the hypothesis. A replication design controls for differential effects of the extraneous independent variables and it helps to randomize any individual differences among those conducting the treatment. The sample is taken randomly from the population and equal number of elements is put in each group so that the size of the group is not likely to affect the results of the study. Randomized block design RBD. In this case, the elements are first divided into groups known as blocks, where elements in each block are given same number of treatments and each element in one block is randomly assigned a treatment. At the block level, the effect of the extraneous variable is held to be fixed so that the contribution to the total variability is measurable. The purpose of such design or randomization is to take care of the extraneous variable and its effect on the experiment. Latin square design LSD. This type of design is usually used to test effectiveness of seeds, fertilizers, etc. That is, this is more used in agricultural studies. Here the field is divided into blocks. And each block is again divided into many parts, but the experimental variable is applied in such a way that each of the stimuli is used in every block. The design presents as many independent variables as there are subjects, but the variables are presented in different and unique order for each subject. Factorial design FD. In simple terms, to see the effect of two factors on the dependent variable, this is the design that is used. In this design, the extraneous variable is called the control variable and the independent variable is called the experimental variable. The sample is divided into 4 cells or groups. Each of the 4 combinations provide one experimental condition. Elements are randomly assigned to each treatment in the same manner. In a more complex factorial design, 3 or more independent variables are considered simultaneously. From this design, it is possible to determine the interaction between each possible pair of variables. Quasi-experimental designs can be of two types: ex post facto research designs, one group post test only design. Ex post facto research design, this design also referred to as the post test only design, where the experimental introduces the test stimulus in the experimental group, or in a most popular version of this design, the test stimulus is beyond the control of the researcher, as he or she comes to the scene after the test stimulus has already occurred. This research proceeds from the past to the present and does not contain any orientation for the future. The researcher can control only one variable which has already been recorded in the past. The aim is to measure a characteristic by comparing two groups similar on all grounds except that characteristic. The researcher will have no chance to pre-test. One group post-test only design. This experiment lacks a control group and the experimental group lacks a pre-test. It has only an experimental stimulus and a post-test. Researchers use this design when they want to measure many variables at the post-test, and if they are familiar with the field situation, they might know what the results will be without a test stimulus. Match study. The most important way to make an experimental group and a control group is to match both pairs on identical subjects such as sex or age. Unless both are matched on important characteristics, the control group and the experimental group cannot be labeled equal. The basic outline of an experimental design is very simple. An experimental group is exposed to assumed causal variable while a control group is not. The two groups are then compared in terms of the assumed effect. The research design based on this simple line of logic depends on three types of evidence to be collected. How to infer based on the evidence collected. Evidence of concomitant variation. The investigator knows which subjects has been exposed to the assumed causal variable. He or she measures all subjects in terms of the assumed dependent variable and then determines whether the dependent variable occurs more frequently among the subjects who have been exposed to the independent variable, show a greater amount of dependent variable than those who have not. Evidence that dependent variable did not occur before the causal variable. It is secured by two ways, one by before exposure to the independent variable. Both the control and the experimental groups did not differ in terms of the dependent variable, and second by measuring the position on the dependent variable before exposure to the independent variable. Evidence on ruling out the other factors. This can be secured in several ways. Among the major ones are identifying the factors that have occurred in the past, contemporaneous variables that other than exposure to the experimental variables, developmental changes that are taking place, etc. The laboratory method. The laboratory method appeals to those who believe that human behavior can be studied and explained in the same way as can events in the world of nature. This is of course a positivist perspective. Despite the attractions between a positivist methodology and experiments in a laboratory setting, experiments can also take place in field settings, that is in real world. Experiments in field settings. Such experiments tend to relax the experimental controls to produce naturalistic conditions. Field experiments appeal to interpretivists because they focus on how the real world is interpreted by people. Field experiments have given us insights about processes such as labeling, stereotyping, and discrimination. Field experiments are attractive because it is seen as easier to generalize the findings and relating them to the real world. There are some issues that are to be taken into consideration in an experimental research. Issues in experimental research. Internal validity refers to the confidence the researcher has that the causal inference from a study accurately explains whether one variable is a cause of another. If the three conditions of causality, 1. Cause precedes the effect, 2. There is an empirical correlation between them, and 3. Relationship between them is not found to be the result of the effects of some third variable on each of the two initially observed. It is said that the causal inference has internal validity. Internal validity is concerned about those things that may affect whether true measurement has been obtained using the measuring instrument. The factors that may lead to problems of internal validity are history, maturation, mortality, instrumentation, testing. The main threat to external validity is that the knowledge that people are participating in a study is likely to impact on the behavior of the research participants. This is known as the problem of reactivity. Reliability in experiments is also of concern to the researcher. If the causal effect of other factors such as a test stimulus ensures, at least in the experimental group, that the post-test score will not be as same as the pre-test, then this can prevent the reliability of a measure. It is imperative that the instrument used in the pre-test and the post-test be rigorously tested prior to the experiment in order to ensure the truth of reliability assumption. The advantages of experimental research. The advantages are opportunity to establish causality, scope for the researcher to control is the ultimate test, to study changes over time. Disadvantages of experimental research. The main problem in an experimental research is the artificial setting. The experimental situation is a reactive method. The social scientist in an experimental study faces a dilemma, placing subjects in a closed artificial setting in order to observe the effect of changes in behavior, and also the fact that an artificial environment may be a hindrance to observing actual behavior. Experimental studies are generally conducted over small samples. The larger the sample gets, the difficult it becomes to generalize on it and also to take control over extraneous variables. Ethical dilemmas in experimental studies. Experimental studies raise ethical dilemmas concerning the manner in which researchers treat people. Some researchers consider it inappropriate to treat humans like animals, manipulating their behaviors in a laboratory setting. Others question the pragmatism of including some and excluding others in the two groups under consideration. Another problem is that in experiments, the researcher is able to establish exactly what it is they will be looking for as an outcome in their research. Experimentation in natural science does not face this problem. Experiments are the best way to prove causality. What we did in this module is to understand how experimental research can be designed. Second, we looked into the various ways in which it can be done, that is, the various types of research designs, experimental research designs. Third, we looked into the issues that were important in analyzing experimental research. Fourth, we sought to find out and evaluated the ways in which evidence is collected for experimental research design. Next, we talked about the ethical issues in experimental research design with the advantages and disadvantages of using this experimental research design. Thank you.